So, what's up class? This is final review. So, do you have any questions you want to go over? Let me know.
Professor, I didn't understand Plasmodesmata. Can you go over that?
That's my favorite topic. Okay, let's go discuss that. So right here, Plasmodesmata. What is Plasmodesmata? You might be thinking. So it's actually a cell junction in the plant cell which communicates between two cytoplasms, which are the adjoining cytoplasms, right? And what's unique about that? You might be thinking. So the unique thing is they can let protein from cells transfer to other cells. Why is this unique? Here we have animal cells like us. We animals, we have gap junctions. They are really small, but they cannot transport proteins. And for this, plants have Plasmodesmata right here, which actually can transfer proteins. There you have it. Okay, who wants to explain how they're formed?
Me.
Okay.
So the formation of the primary Plasmodesmata occurs during cell division. To do this, endoplasmic reticulum, it positions itself in the cell plate. This means it is now present in the mother and daughter cells, in both mother and daughter cells. Kinesins, it walks along microtubule to continue deposit material for cell wall around endoplasmic reticulum. So the next step is the actin filament coiling around endoplasmic reticulum, which forms a desmotubule. This allows for more complex channel structures and regulation of the width of desmotubule. Oh, Professor, I don't know what will happen after this when it comes to transport. Could you help me?
Yeah, for sure.
Thank you.
Man, move, man. I'll talk about transport. You can transport your ass out of the classroom. That's my turn. Transport through the Plasmodesmata is very easily done. The Plasmodesmata has a very wide range, having a channel size of about 40 to 60 nanometers. Because of this large size, small molecules like sugars, amino acids, and proteins can pass freely through. And the most obvious molecules like ions and water that pass through in any cell, even animal cells, they'll also pass through very easily. But the one that I want to highlight in this case is proteins, because in many parts of the animal kingdom, proteins have to be shipped out through cells through vesicle systems. But plants make it a bit easier using Plasmodesmata. Proteins can be marked for shipment using these three methods. One could be amino acid sequence. So similar to how we see nuclear localization sequences in proteins that ship proteins to the nucleus to act on DNA. Instead, we can have amino acid sequences that cause it to be shipped to the Plasmodesmata and pass right through. Another one could be chaperone proteins, where they would connect to another protein and bring it across the cytoplasmic channel. And the most interesting one to me would be from ER to ER shipping. So the proteins that are made in the endoplasmic reticulum of one, the first cell, would go through the lumen and end up in the other cell, through the desmotubule. And this is very interesting to me because it doesn't require any extracellular transport.
Sarbjot, that was a nice explanation, but that behavior of yours was highly inappropriate. So, apologize to him right now.
All right. Hey man, look.
Whoa, 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 whoa! Security! Security, take him to the dean's office! What brings you to my office?
Sir, take a look at this.
Man, move, man. I'll talk about transport. You can transport your ass out of the classroom. That ain't me.
What is that?
I'm sorry, sir.
No,
but
what is that?
I'm sorry. I didn't mean to.
No, you're not understanding. What is plasmodesmata?
What is a plasmodesmata?
Yeah.
I'll be happy to show you. So what a plasmodesmata is that it transports molecules through two cell bodies.
3 hours later.
Oh, we see you're back. I hope the dean gave you a good lecture.
Yeah, really, I gave the dean a lecture.
Whoa! You give the dean a lecture. Okay, come up to the board and actually lecture us, Professor.
Okay, I guess you'll learn something too. All right then, listen up. So I'm gonna be going over the mechanism of how plasmodesmata operate in the plant cell. So the first thing I want to cover is SAM organization and how it's done through plasmodesmata. So WUS is only expressed in the organizing center, and it has to move to the central zone in order to create CLV3. And the way it moves is through plasmodesmata. And in the specific way, it uses a chaperone protein to guide its way to the plasmodesmata and through over to the central zone. In the same way the WUS protein moves through plasmodesmata, the auxin hormone does as well. But instead, it does this in response to light. So initially in a dark environment, the plasmodesmata is open to all, allowing free passage of molecules in both sides. However, when light is shown on the plant, it induces GSLA expression, which then induces callose synthase expression. This piles up callose next to the neck of the desmotubule, restricting all movement of molecules. And what this does to auxin is that auxin is built up in one side and it can't move to the other side, thus causing an auxin gradient, which then results in phototrophism.
Sarbjot, that was really nice. Really good.
Thank you so much.
But don't misbehave next time.
All right,
Okay?
You're the boss.
Right here, Sarbjot has actually explained us all the mechanism about plasmodesmata. So let's go to pathogenic modulations for plasmodesmata. So first, let's discuss viruses. So virus, they make movement protein. And you know how movement protein actually work is the first thing, they actually combine with RNA and they form a complex which can easily transport itself from the plasmodesmata. The second thing they can do is that the movement protein actually transform into tubules, and these tubules are very small and they can actually pass through the plasmodesmata, right? And the third way, which is one of the most important ways to understand, is that they can alter the architecture of the plasmodesmata. So what they do is they disturb the actin filaments, and when actin filaments are disturbed, the plasmodesmata pore size it increases, and once it increases, the virus can go through. Let's discuss the fungi. Now, in fungi, the main role is that they inhibit the callosynthase. And you know, callosynthase actually makes the callose. And if there's no callose which is being formed right there, then the pore size is going to increase and the fungi can actually move. Now you might think the host is under attack, but sometimes we have observed that the host actually has this defense mode in which it actually activates the callosynthase, which makes the callose so it constricts the neck and henceforth the pore size decreases. And at the same time, it inhibits the beta-1,3 glucokinase, which actually degrades the callose. So obviously it's inhibited. It's not going to degrade the callose. We have callose on there, and there we go. The pore size still decreases. So that's the pathogenic modulation of plasmodesmata. So there you go. We have learned everything about plasmodesmata. Come up, class. Tell us what you actually learned today.
I learned about transport.
I learned about the viral modulation of plasmodesmata.
Okay, class. So the thing is, we have the final, which is 200 questions in 20 minutes, and it's not curved, and everybody dropped the class, and it's only you two.
Man, what the heck, man?
Hey, Sigoni, hey!